Hey guys, today's learning target and lesson is going to be all about how stuff gets in and out of the cell. So how do molecules move through a semi-permeable membrane? How do we get things in and out of the cell and transport them around? So to begin with, we'll talk about two major categories. There's passive transport and active transport. Passive simply means that there's no ATP used in the process of moving particles or molecules in and out of the cell. We have three ways that this happens. So the three ways are going to be diffusion, facilitated diffusion, and osmosis. So we'll start with diffusion and talk about this. There's a concentration gradient, which simply means that there's an area of high concentration and an area of low concentration. So when looking at this video here that I'll play in a sec, you're going to see that molecules tend to move from an area of high concentration to low. The natural order of things is establish equilibrium or balance, and it's going to continue this process until that's occurred. So we'll take a look at that real quickly and see if we see this movement of an area of high concentration to low. So we should see this. These orange things could be molecules or particles, and they're going to continue to move through those rectangles, which we could call our cell membrane, until there's some kind of balance. So this will stop just short of it, but you can see there's a tendency overall to get movement from down here in the high concentration area up into the low concentration. The other two modes of passive transport would be osmosis, which is a diffusion of water. It's exactly the same as that other category, only talking about water moving from high concentration to low concentration through a membrane. And then the final one is facilitated diffusion, which is a little bit different. This is purple guy here that's grabbing these orange molecules and moving them into the cell from the outside of the cell. It's just really a protein that kind of binds with these molecules and changes its shape. And when it changes its shape, it kind of stuffs it inside. But it doesn't require the use of energy. Here on the bottom you can see this picture, and then we'll go back to the top. This is active transport. This is our other category. These ATP molecules, these big yellow ones here, bind to this purple protein and they give it energy to move. What they're doing is they're moving things against the concentration gradient at times, and that's going to allow that energy is needed to push those things in the opposite direction. So you saw the yellow ones moving towards more yellow ones. That would be our sodium, and you saw potassium moving towards more potassium. So they're moving from a low concentration to a high concentration. They're going against the natural order of things, the gradient. They're going against equilibrium, and when they do that, we need energy to force it in that direction. It takes energy to move things in that direction. The other couple ways that we'll talk about how energy is needed is endocytosis and exocytosis. Endocytosis, endo means into the cytoplasm, a process where the cell will take in a foreign substance. So here we can kind of see this. We don't have an animation here, but we can see this cell membrane phospholipid bilayer kind of surround this blue particle. It creates a vesicle here, and the vesicle breaks apart, and so does the compound whatever it is, the particle can be used inside the cell. Exocytosis is exiting the cell. So here's the cell membrane. You can see these vesicles are made of phospholipid bilayers. They kind of fuse right here, and then they push things out of the cell. There are three major types of solutions that cells can be exposed to, and you kind of see this animation at the bottom too. We'll show you this. There's isotonic, hypertonic, and hypotonic. Isotonic is a solution that really allows water to flow equally in and out of the cell, as well as other non-water molecules. And isotonic solutions, when cells are in them, remain at equilibrium. There's balance. There's equal amounts of particles in and out of the cell. So the concentration of non-water molecules outside the cell is the same as the concentration of non-water molecules inside. Equal water in and out of the cell. Cell keeps its shape, and water continues to undergo that process of osmosis in and out of the cell. The next type of solution is a hypotonic solution. A hypotonic solution is when we have a high concentration of non-water molecules in the cell, or there's less water in the cell. You can think of it that way. Low concentration of non-water molecules outside the cell, or more water outside the cell. So in this case, you can see the water molecules are higher out here, So what's going to happen is osmosis will cause the water to move into the cell. The water's too crowded out here. There's too many of them, and they're going to flow in the direction of the cell across the yellow lines in the cell membrane. So we definitely get a net flow of water in this direction overall. The cell's going to get bigger if we do that, and we allow the cell. We can see a cell here starting to get fatter, swell. The cell can explode or burst if water continues to rush in this direction. The next solution, the last one we'll talk about here, is a hypertonic solution. This is when we have a high concentration of non-water molecules outside of the cell. So there's less water outside, there's more water inside. What's going to happen if there's more water inside is we get a net flow in the other direction. See all these water molecules out here? The smaller ones are water, and what happens is those water molecules right here is the start of our animation. We have these water molecules that are going to start to flow in this direction from inside the cell to out, and that will cause the cell to shrink or shrivel. Osmosis does occur in plant cells too. Two important ideas here is that osmosis in plants allows this turgor pressure to be created, and then turgor pressure builds up inside the cell and helps the plant stand up. It presses against the cell wall, and that's really what our vacuole is doing, is providing that pressure when it swells with water and allows that cell wall to stay rigid and the plant to remain upright. Plasmolysis is the idea that water can result in a drop of turgor pressure when it starts to go away, and the plant will eventually end up wilting, and that's kind of this plasmolysis process, is the wilting of the cell.